Hello everyone, welcome to my YouTube channel. This is Enoch's tutorial, so don't forget to like and subscribe to watching the video so you can receive every bit of updates from this channel. In today's video, we're going to look at the structure and part of chromosomes and also how chromosomes are formed in the eukaryotic cell. So what then are chromosomes? So chromosomes are basically found in the nucleus of a cell, basically the eukaryotic cell, that are made of a DNA strand and a protein molecule called histone. So chromosomes are made of two main components. We have a DNA strand that coils around several times a protein molecule called histones. It becomes visible when there is a need for cell division. Chromosomes are regarded as genetic material because they are made of DNA, and DNA are made of sections that code for particular proteins which define the feature or the trait of an offspring from a parent, which you call a gene. So a gene is a section on a DNA that codes for a particular protein that is very distinct among different individuals, or let's say different species. So how then are chromosomes formed? So chromosomes are formed through several stages. They are formed through several stages. In most cases, chromosomes become visible when there is a need for a mitotic cell division, but in the nucleus, it normally exists in the condensed and invisible form. So chromosomes are formed basically when there's a coiling of a DNA strand around a protein we call histone. So when a DNA strand, which is thread-like, coils around several times around a protein we call histone, we form what we call a nucleosome. So here, the first stage of chromosome formation is that the DNA strand coils around a protein molecule called histone to form what we call a nucleosome. Now there is a further coiling of the nucleosome like this. So here the first one is the DNA strand coils around the protein molecules here as shown here. So when they coil around this protein, we form what we call a nucleosome. So these threads are what we call the nucleosomes. So nucleosomes further coils several times. So when they coil several times, they form what we call a chromatin. So chromatin is invisible in, or it is condensed in the nucleus. Chromatin is invisible, it's condensed in the nucleus. It only becomes visible when there is a need for cell division. So when a DNA strand coils around a protein, they form what we call a nucleosome. Then the further coiling of the nucleosomes, as shown here, forms the chromatin. So the chromatin is the invisible form of chromosomes, or the chromatid. Let's come there. Now, when there is a need for cell division, that is when the cell is about to divide, the chromosome must split up. That is formed by first, the chromatin, which is in the nucleus, become visible. When they become visible, they form what we call a chromatid. So this is a chromatid, which is a visible form of the chromatin. The chromatin is formed from the coiling of the nucleosome, which is also formed by coiling of the DNA strands around the histone protein. So when they become visible, then you have two strands of chromatids. So this chromatid is made of a protein histones that has been intercoiled several times by a DNA, or it is made of a nucleosome that has coiled several times. So this chromatids is just one segment of a chromosome. So if this is a chromatid, two chromatids come together to merge at a point, which forms two chromatids. So chromosome is made of two chromatids. Each of these are what we call sister chromatids. This is one sister chromatid, and this is another sister chromatid. They are joined at a point we call a centromere. A centromere. So basically, DNA, or let's say chromosomes, are made of two components. It is the protein and the DNA. But when they form the chromosome itself, they get joined at a point. Let's say two sister chromatids, which is a condensed form or visible form of the chromatin, get joined at a point we call the centromere. So the joint portion of a chromosome is called the centromere. The centromere is the point where the two sister chromatids in a chromosome joins. And the point of attachment, or what joins the two chromatids together, are what we call the kinetochore. The kinetochore is just a segment or is a protein or a molecule that joins the two chromatids together to form what we call a chromosome. And the section of the joining is called a centromere. Take note of this. The section of the joining is called a centromere, whereas what joins the two sister chromatids is called a kinetochore. Now, this is a chromosome structure, and this chromosome has several parts. Aside being made of two main components, that is DNA and a protein histone, the chromosome has several parts. It can have the kinetochore as we discussed here. It can have the centromere as we discussed here. It can have a lower arm, or let's say a shorter arm, that is the shorter portion of each sister chromatid, and also a longer arm. Each tip of the chromatids, the tips of each chromatids are called a telomere. The telomere is the tips of each chromatid. So this side, this side, this side, and this side are called the telomere of the chromosome. Once again, the tips are called the telomere. The longer segment from where the kinetochore is called the longer arm. The shorter one is called the shorter arm, whereas the point of joining of the two sister chromatids is called the centromere, and what joins the two chromatids together are called the kinetochore. So let's take a look at this structure. So as we said earlier, chromosomes only become visible when there is a need for cell division. When there is a need for cell division, the spindle fibers from each pole of the cells grows and attaches to the kinetochore of the centromere at the centromere. So this is what joins the two sister chromatids together, and the point of the joining is called the centromere. The kinetochore is the point of attachment of the mitotic spindle fibers. The kinetochore is the point where the spindle fibers attaches and split it apart during mitotic cell division. Whereas the constriction point of joining of the two sister chromatids are what we call the centromere. So here, the top here is the top here is the shorter arm, the longer here at the lower arm. These molecules that join the sister chromatids together are called the kinetochore. The constriction point of joining is called the centromere, and the edges or the ends of each sister chromatid is called the telomere. Let's look at this. So we have here the centromeric region, that is the region of constriction of joining. We have the kinetochore here, that's where the spindle fibers bond or binds to split the protons, the chromosomes apart during cell division, and also the sister chromatids, that is from the visible form of the chromatin after coiling of the nucleosomes. That is how basically chromosome structures look like. It has a telomere, which is not indicated here. It has here the telomere. We're going to indicate that. It has here the lower arm, the longer arm, the lower arm, and also it has the centromere. So let's briefly go through how chromosomes are formed. So as we discussed earlier, chromosomes are formed when or it's a DNA molecule coils several times around a protein molecule called histones. So this is how chromosomes are organized in an eukaryotic nucleus. The first one is the DNA double helix. The DNA double helix coils around a histone protein like this. The net product is called the nucleosomes. The nucleosome. So this is the nucleosome as shown here. When DNA molecule coils around the histone, you form what you call the nucleosomes. Then the nucleosomes coils several times. So when it intercoils several times, as shown here, these strands are the nucleosomes. When the nucleosome, which is made of the coiled DNA around the histones, also intercoils several times, they form what we call a chromatin. The chromatin is just condensed and is invisible in the nucleus of an eukaryotic cell. But during cell division, the chromatin becomes visible and further condenses to the visibility is promoted by the further condensation of the chromatin, which forms what we call a chromatid. And two chromatids joins at a point here to form what we call a chromosome. So the chromosome is what is seen during cell division. That's a summary of how chromosomes are formed. So let's look at the parts of a chromosome once again. So the first one The tips of the chromosomes are shown here. The tips of the chromosomes are shown here are called telomere. Telomere. So being upper or lower, this segment too is called the telomere. So the telomere is the tips of each sister chromatids in a chromosome. Telomere as in Telomere, telomere of the chromosome. This part, the shorter part from the edge to the point of constriction, is called the lower or the shorter arm. The longer one is called the longer arm. Now this point of constriction, the whole of this point of constriction is called the centromere. The centromere is the point of constriction of the two sister chromatids. In the centromere, each chromatid is joined by a molecule we call the kinetochore. So the kinetochore is this whitish thing here, which joins the two sister chromatids together. So these are the parts of the chromosomes. Thanks for watching. Don't forget to like and subscribe for watching the video so you can receive every update from this channel. Thank you.